last time we covered section 4.2. There's one piece from that section that I forgot to do. Didn't realize that I had talked about it, and that is margin of error. So I'm going to do that first. And then once we get margin of error defined, we'll move on into section 4.3 and do that section today. So margin of error is a term that you've probably heard at some point in your life when you watch the TV news reports of political polls and things like that. And this is a good time to be having this discussion because we're in the middle of the midterm elections, right? And what are we, 5 or 6 weeks out from election day? So there's been a lot of political polls going on lately. So when you hear a political poll and they say something to the effect that 36% of the registered voters say they're going to support candidate John Doe, and that is a margin of error of plus or minus 3%. What the heck does that mean? Margin of error.
The actual results can be different by 30%.
Okay, you said a couple of good things there. The actual results. Okay, what do you mean actual results as opposed to the results of our survey?
Their survey didn't catch all possible answers.
Okay, good. The survey is not going to be exactly correct. We know that. We know anytime you do a sample from a population, your estimates from that sample aren't going to be perfect. And what you're always trying to do is estimate the truth in the whole population. So in this case, the truth is out of all those people who actually go vote, what percent of them support candidate John Doe? Well, our survey is not going to hit that exact. So there's going to be some error. The margin of error, now what was the second part of what you said?
I don't remember now. They're off by a certain percent.
You will be off by no more than that amount. Okay? So sample estimate will be in error by no more than the margin of error. So if you say 36% with a margin of error of 3%, then you're saying, well, our sample had 36%, but the truth is probably somewhere between 33% and 39%, plus or minus 3%. One of the questions on the test had a deal with margin of error. It said the margin of error was 4, and from that they estimate that the population mean, it was a mean, not a proportion, but the population mean is between these two numbers. And if you notice, those two numbers were 4 units on either side of the sample mean. And that's the way a margin of error works, right? So for proportion or a percent, it's the sample proportion plus and minus the margin of error gives you an interval in which you are pretty sure that the true population proportion is in there. Now the way you estimate margin of error, in this chapter, they give us an approximate margin of error. Now this is my own little abbreviation for margin of error. I call it the MOE. Right? Margin of error. M-O-E. In the book, I think he just uses an M. But M can mean median or mean or whatever, so I try to distinguish that. And a fairly good approximation to a margin of error is just 1 over the square root of the sample size. And then you multiply by 100 if you want to write it as a percent. You leave the 100 off if you want to leave it as a proportion. Most of the time we talk in terms of percent, so he multiplies by 100. Okay? So what do you notice about this as related to the sample size? As the sample size gets bigger, what happens? The margin of error will decrease. We've got n on the bottom of a fraction. If you make the bottom of a fraction big, you make the value of that fraction small. and vice versa. So one of your homework problems tonight, at least one of them, has you compute an approximate margin of error, and this is what they're asking you to do. And I think they have you do it for several different sample sizes. So if I had a sample of size 100, what would my approximate margin of error be? Well, let's see. The margin of error would be 1 over the square root of 100 times 100. 1 over square root of 100 is 1 over 10, which is 1/10. So 0.1 times 100 is 10. So the margin of error would be 10%. Now if you think about it, 100 sounds like a pretty big sample size. But what this is saying is, if you use 100 people in a survey or a sample to estimate a proportion, then you can be no more confident in your answer than plus or minus 10%. So if I do a survey of 100 Washburn students and I say, well, in my survey, 60% of them approved of this new ordinance, whatever it is. Can I say conclusively that a majority of students support it?
No.
No, because the margin of error would be 10%. So the truth is, all I can say is, well, it's somewhere between 50% and 70%. And 50% is the line for majority, and it might not be a majority. It might be exactly half. So if I want to be more sure, if I want to narrow that range, be more precise, what do I need to do? Get more people. That's right. Get a bigger sample size. I think in the book they then have you go to n equal 1,000, or I'm sorry, in the online homework. If I make that 1,000, what happens? You have to do it on your calculator because 1,000 is not a perfect square. What's the margin of error if you have 1,000? One over 3.1? 3.16. Okay? So 1 over 3.16 times 100. Now on your calculator, you can either do 1 divided by 3.16, or you can do this all out in one big step, or there's a button with an x to the minus 1 power. But what do you remember from algebra class? Something to the minus 1 power is? That's the inverse, the reciprocal of it. So this is the same thing as 1 over x. So if you want the reciprocal of this, 1 over it, you just do x inverse. But either way, what's that turn out to be?
It's 3.16 is the answer.
Oh, 1 over?
It's 1 over 0.0316.
Oh, okay. Well, doggone it. You're too fast for me. You did too many steps. 1 over point what?
0316.
0316. That looks better. And when you when you take the reciprocal, multiply by 100, you get 3.16. So if you get 1,000 people, now your margin of error is about 3% instead of 10%. So you really narrowed it down. Most surveys, like political polls and things like that, will try to get their margin of error down to 3 to 4%. You rarely will see one in the media with a margin of error more than 4%, which means they had to use a sample size that was pretty darn big in order to do that. Okay, any questions on margin of error? It's actually, this, like I said, this is only an approximation of the margin of error. The actual formula we'll learn in, I think it's chapter 7, and it's a little more complicated, but this is pretty close. Yeah, Marianne?
Sorry, just in case somebody were to get confused, it's actually 1 over 31.62.
Thank you. 1 over 31, yeah. Yeah. This, I'm not paying attention to what I'm doing. What I harped on you, I said, always look at your answer, see if it makes sense. Square root of 1,000 being that doesn't make sense, right? So this is 1 over whatever square root of 1,000 is that fraction. So I keep just listening to what you say and not paying attention. It's 1 over what?
31.62.
31.62. Someday we're going to get that right. And when we do, we'll have a party or something. Okay. we're ready for section 4.3. And 4.3 is titled, "What are good ways and poor ways to experiment?" So, boy, there's that word experiment again. Let's refresh our memories. What's an experiment?
You separate two groups randomly, and then
You're describing a good experiment. She said you're going to separate two groups randomly and then
do your experiment on them. Like, one's a control group, one you do something to.
Assign treatment.
okay, you assign some kind of a treatment, right? And that's the hallmark of an experiment, is you impose some kind of a change on the subjects, a treatment or whatever. But you impose it. You don't let them self-select to that treatment. Now, what Chelsea described was she threw in several words. You can tell she's already read the section, which I like, or at least you've heard of these terms before. She used control group. She used randomly assigned. Those are good ways to experiment. But it doesn't have to have those good things in it to be called an experiment. Okay? Here's an example. This is out of the literature. This is not a real up-to-the-minute article. I downloaded this a couple of years ago. What, you had a question?
Yeah, randomly I'm trying to figure out, like, would this be random, like, if you just split the group down the middle? That's not really random.
If you split the group down the middle, no, technically that's not random. Now, we do some of those kind of things in research sometimes thinking, well, it's pretty much random where you sat in the room. So if I split down the middle, that's about as random of a division as, using my calculator and whatever. But technically, no, that's not random. But if you say, no, smart people tend to sit close to the teacher and dumb people tend to sit, if you had some suspicion like that, then you wouldn't want to just split the room, right? Of course, that's not the case. It's people that are tired, they want to be in the back so they can sleep. Right, Eddie? Mm-hmm. Now, let's look at this article real quick. As quick as I can get it up anyway. There. Melatonin agonist shows benefits in treating bipolar depression. Okay? I'm not going to get into too much of the detail about a melatonin agonist. Basically, it's a drug that they're going to give them and try to treat their bipolar depression. Okay? So let's scroll down. There's a lot of kind of intro stuff. This is a newspaper type article. It was in the Medscape Medical News from 2005. This is not a journal article, but this study was published in a journal as well. Here I highlighted in yellow the paragraph that kind of talks about the design. Therefore, in an initial study, Drs. Gulfi, sounds like goofy, but Gulfi and Calabrese recruited 21 patients with bipolar disorder who were experiencing a major depressive episode to see if adjunctive treatment with agamelatonin, in addition to a mood stabilizer, would improve their symptoms. And then it talks about, what instrument they used to measure the subjects, how they got their response variable. So the response variable was this Hamilton rating scale for depression. Right here, it says all patients received 0.25 milligrams of agamelatonin. However you say that. Is this an observational study or an experiment?
Observational. Neither.
They're imposing
It's an
that treatment, they're
observational
imposing on
experiment.
everyone.
What did you say, Jory?
I said it's an experiment, but it's a bad experiment.
It is an experiment, but it's a bad experiment. That's exactly right. It's an experiment because they imposed a change on the subjects. The subjects came into the study, they had never taken this drug, and then they said we put them all on 0.25 milligrams of this drug. So we imposed that change on them. That, by definition, is an experiment.
There's just no control.
But there's no control group. It's not a good experiment, there's no control group. Right?
There's only one
Now-
type of medication, or there's only one.
Right, there's only one treatment used in the study. Okay, so now that you understand that it doesn't have to be a good experiment to be called an experiment, let's talk about some of those terms that we just threw out because they're all definitions that are presented in this section. First of all, we said control group. What is the control group?
People you don't do anything to.
No treatment.
I'll accept that for now, but we're going to revise what he just said. He said, people you don't do anything to. That's not quite right. We're going to do something to them.
You give them
Yeah.
It's the people you just observe, you just give them a placebo type thing, you just observe.
Okay? Yeah, you just threw out another term we're going to need to get down. Placebo. Okay? And the answer is no, you don't just observe those people. The control group is a group that does not get the experimental treatment. Now, a good experiment will treat the control group exactly the same in every other aspect. If I write a protocol for a clinical trial and I say that at the baseline visit on day 0, I'm going to bring them in, I'm going to draw this much blood, and I'm going to test for this, and I'm going to measure symptoms using this depression scale or whatever it is, that I'm doing that to my control group too. Everything identical. The only thing different is in what treatment they got. Okay? Now, you said, or before I get to this, why is it important to have a control group?
So you can measure results against.
Change it.
Yeah.
I heard about 15 people talking once and I didn't get any of it.
Comparison.
For comparison, exactly. This allows comparison. And I'm going to put the word concurrent in there. Concurrent comparison. Now, what the experiment that we just looked at did was they did not do concurrent comparison. What they did is said, we measured their depression scale, then we put them on this drug, then we remeasured. So they compared each person to their previous baseline depression score. So they had a comparison, but they didn't have a concurrent comparison. What a better experiment says, well, there could have been things that happened in the intervening time that changed their score besides the treatment. And unless you have a control group to pick that up, you'll never know. A little easier example to understand, I told you before that before I started teaching at Washburn, I worked in the pharmaceutical industry for quite a while. And one of the drugs that I worked on, I was the statistician for Allegra. Have you heard of Allegra? It's an allergy medication. It's on the market now. And I was the statistician involved with getting that drug approved and onto the market. And the way you study allergy symptoms, or an allergy drug, is you give them the drug and you have them rate their symptoms. And you know, that's a pretty subjective thing, right? How are your allergy symptoms today, Delaney?
Terrible.
Terrible. So on a scale of none, mild, moderate to severe, where would you put them?
Moderately severe.
Moderately severe. See how she didn't use one of the things I tried to get? And you see it's kind of subjective, right? Well, suppose I did an allergy study for Allegra, and I did it like the one we just looked at. I didn't have a control group. Is there anything that you think could happen in the intervening week? I have them rate their symptoms at baseline before I give them any medication. Moderately severe. Now, okay, well, here's some pills. Take these home. Take them for a week. Come back. We're going to rate your symptoms again. And she comes back. She says, ooh, I feel a lot better. I'm down to mild symptoms, right? Is there anything besides a true drug effect that could account for that?
Her allergy, what she's allergic to.
Okay. The pollen count. I heard change in season, the outdoor environment, the pollen count. If it goes down during that week, then sure, she's going to look like she got better. And so will everybody else. If I have a control group, guess what? They get better too. And then your comparison says, well, you're no different than the control group. Yeah.
So assuming that a researcher is not trying to make a particular point with this study, why would they ever design it like without a control group or just poorly in general?
That's a great question. Did everybody hear her question? She said, well, assuming that the researchers are unbiased, and that's, sometimes they are, sometimes they're not, but why would they ever do a bad experiment? Well, that's if they're unethical. He said get the results they want, right? But she's saying assuming they're not a biased investigator, why would they ever do bad experiments? Sometimes you just can't do a good one, right? There's just no way to do everything that's a perfect experiment. It's too difficult. So you do the best you can. If you go on and read that whole article about that study, it was like an early phase study. I'll talk about phases in a minute, but let me jot that down so I don't forget. Where you might do an experiment that's fairly quick, fairly easy to do, doesn't take very many patients, and you're just looking to see, is there some hint that this treatment might be effective? And if there is, we'll go on and do a much better, well-designed, well-controlled clinical trial. Okay? So that's kind of the reason sometimes those kind of studies get done. But sometimes they catch the media's attention like this one did, really prematurely. Because a lot of times when they get to phase 3, they're like, it didn't work. We thought it would work, but it didn't. Because when we did a control group, the control group got better also. Okay? So that's control group. Very important that you have a control group. And it's very important that you treat the control group exactly the same as your experimental group, which a lot of times involves placebo. So let's define that. What is a placebo?
Sugar pill.
Sugar pill. I was hoping somebody would say that. It's actually not a sugar pill. I heard you say fake drug, right? Which is a little more accurate. Most people, when they hear placebo, they think sugar pill, and that's what they'll even say in the media. In fact, nowadays they do those what they call direct-to-consumer marketing ads on TV. Those ads about prescription medications where they do this laundry list of things that this drug can do to you. It's like it can make you grow a third toe, or whatever. And a lot of times they'll say, when compared to sugar pill. And they say it that way because the general public doesn't necessarily know what placebo is. But I guarantee you it was not a sugar pill. Right? And why is it not a sugar pill? Well, it's because when patients get into a clinical trial, they have to tell that patient what's going to be happening to them. It's something called informed consent. Have you heard of this term? Informed consent. You can't just put somebody in a trial and not tell them what you're studying. And especially you can't tell them that you can't not tell them that you're doing something to them, like giving them a medication or whatever. Right? They used to actually do studies that way once in a while, but the government caught on and passed some laws, and now by law you have to have informed consent before you can legally experiment on humans with drugs or diets or anything. Informed consent. Well, if they sign the informed consent, it says in there there's a 50-50 chance that you get active medication or you get placebo. So they come in for their visit, they get randomized to treatment, they're handed a bottle of pills, they go home, guess what the first thing they do is? They try to figure out what group they're in. And what's the way they try to do that?
Taste it.
They taste that pill. They pop one in and they bite it. You're supposed to just swallow it, right? And if it's active medication, what's it probably taste like? Nasty, bitter, horrible, yuck. If they bite it, it tastes like sugar. Then they know they got placebo, right?
You just make a bitter sugar pill.
So that's what they do. Most big pharmaceutical companies have a whole department of people whose job is to make placebos for their clinical trials. And the placebo should look, taste, and smell identical to the actual medication, so that there's no way to tell that it does not have the active ingredient in it. Okay. Now, why is that important? Why is it important that they don't know it's placebo?
That'll affect their,
Placebo effect.
What'd you say?
Because of the placebo effect.
Because of the placebo effect.
that.
Okay,
Like-
let's define that. What's placebo effect?
Their perception of the effect of the drug. Like how they,
Okay.
whether their mind's gonna tell them, oh, well, this isn't gonna work because it's fake. So whether it's real or not, it's gonna
The
have-
placebo effect is when patients improve because they think they are getting treated. Simply by the fact that their mind thinking, oh, I'm getting a new medication, I must be better. They actually do improve. Now, in allergies, in a study like the Allegra trials, you can see how placebo effect can come into play, right? If I got sent home with a bottle of pills and told to come back a week later and rate my symptoms, if I knew that I was on placebo, I might say, well, you know, I feel a lot better this week, but I don't want to look stupid. So no, I haven't changed, right? But if I don't know it's placebo, then I might say, yeah, I feel better. So knowing whether it's placebo or not can affect how they answer the question. Right? But also, you can imagine how, you know, something that's as subjective as symptom ratings in allergies, that you might think, well, yeah, I guess I do feel a little bit better. But placebo effect is actually much stronger than that. It happens in disease states where you might not think it could happen. For instance, another drug that I worked on for a long time was an anti-convulsant. It was for epilepsy patients, right? Stop seizures. Now you wouldn't think that there could be a placebo effect in seizures, but there is. There's a fairly strong, well-documented placebo effect. They're not really sure why, but seizures are basically electrical short-circuiting in the brain, and stress can aggravate that. So, you know, having seizures and waiting when's my next seizure going to happen is pretty stressful. So you get them into a clinical trial where you're seeing them, the doctor's seeing them regularly, and they're getting a new medication. They kind of relax, the stress goes down a little bit, and their seizures slow for a little while. Unfortunately, it's short-lived. It comes back, but there is a placebo effect. So you have to give a placebo to your control group. You have to give something. It doesn't have to be a placebo, but you have to give something to your control group. Otherwise, you can't estimate the placebo effect. Okay? So let's go back to my Allegra trial. You guys have convinced me now that I need to add a control group. So I add a control group, but I don't give them anything. Right? And lo and behold, by the end of the trial, on this diagram, up is good, right? So this is improvement in symptoms. So here's baseline. Both groups are going along and they're at about the same average baseline. And I give my treatment group treatment and they jump up to here. Their symptoms improve that much. Okay? I don't give anything to my control group, so they don't change. That distance is what we call our treatment effect. But if I don't give placebo, is that true treatment effect? Or is it all placebo effect? Or is it some combination?
Yes.
There ain't no way to know. Right? There's no way to know if some of that's placebo effect or all of that's placebo effect. There's just no way to know. So you give placebo to your control group and they jump up to there. Let me leave that line there. This is the overall improvement in symptoms. This much is placebo effect. This much then is the treatment effect. And you can mathematically subtract out the placebo effect and say our treatment had this much improvement above placebo. You can estimate the placebo effect, you can estimate the total effect, and you can estimate the true treatment effect. Okay? Now, we've already talked a little bit about why it's important that they don't know they have placebo. because it can affect the way they rate their symptoms, right? If the patient doesn't know what they're receiving, then we call that a single-blind trial. Or single-blind study. The patient is unaware, but investigators know. And I'll put the word investigators in quotes. That's kind of a generic term. It's the physician that's treating the patient. It's the nurses involved in the study. It's the study personnel at the site. Whoever's involved, they're called the investigators. Okay? So if the patient doesn't know, but the investigators do, that's called single blind. Okay? That's better than what's known as an open label study. An open label study simply is no blinding. I recruit Delaney to be in my study, and I said, Delaney, here's a bottle of pills. I want you to take one of those pills every day for the next week, and come back in, we'll measure your symptoms. Oh, by the way, that's placebo. What's she going to do with that bottle of pills? Throw it away. She's probably going to throw it away, right? So it can bias the study just because I lose that patient. Right? Yeah, question?
What kind of study would you use in an open label study?
Good question. Open label study. That's kind of the other question we were getting. Why would you ever do a study like that? Well, here's where open label happens most often. Life-threatening illnesses that are difficult to blind treatments. Think if you had a cancer study that you're doing. Is placebo ethical? You can't give placebo to somebody who's going to die if they don't get treated, right? So we do what are called active control studies, where you get randomized to either go on to the already approved, commonly used chemotherapy, or you get randomized to our new investigational chemotherapy. You get one of those two. Right? And those almost always are done as open label studies. They know which one of those two they got randomized to. Right? And a lot of the reason that they know is because physicians are not willing to put patients into a trial if they don't get to know what that patient's getting, if it's going to, if it's a life-threatening illness. If it's an allergy study, sure, you're not going to hurt anybody. Right? But if it's a life-threatening illness, a lot of times they're done as open-label studies. And a lot of times it's, I want to compare this chemotherapy to surgery, and I don't know which is best. Well, there's no way to blind that. Right? Well, actually, there are a few examples in the literature of what are called sham surgeries. The most famous example is in Parkinson's disease, where they had they do surgery in the brain and they implant these electrodes. I don't know if you know Michael J. Fox, right? He had this surgery done. Right? Well, the trial that showed it worked, the patients were willing, through informed consent, to sign up and say, it's okay for you to cut my skull open and then sew it back shut and not do anything if I get randomized to that group. Because that's how important it was to do good research and answer this question. They were willing to do that. So that's sham surgery, but most of the time you're not going to find patients that are willing to do sham surgery, so you can't blind it. You either got the surgery group or you got the chemo group. Okay, so what's a double-blind study?
Neither investigator nor patient knows.
Right, neither knows. Neither investigator, I'm going to do some abbreviating, or patient knows. Now, why might it be important to blind the investigators?
You might tell.
Okay, they might tell, they might let it slip, but they might treat them different. That's the main thing. You can bias the results if they treat those patients differently. If the physician knows, well, this patient's on placebo, I really ought to be doing something else for them, and they give them different stuff that's not part of the trial, You just bias the study. Or particularly in the case, a lot of studies, almost all studies actually, will have symptom ratings done not only by the patient but also by the physician. They call them like physician's global assessment. As the physician, how do you feel this patient is doing? Right? Well, if they know that's a placebo patient, they may answer that question differently. So it biases the study. So double blind. Okay? Now, if neither the physician nor the investigator know what drug they're on, who does know?
The person doing the experiment.
Okay? The sponsor of the experiment. Usually it's the statistician that runs the randomization code and then keeps it locked away in a protected server with a password or whatever that nobody knows. And I remember I talked the other day about a study I was actually working on, and it came the day for unblinding. Well, that's when we unblind the study. We attach the randomization code and say, oh, this patient got active, this patient got placebo. When I was in graduate school, I worked as a student intern in a little company in Fort Collins, Colorado, that this was a dental products company. And they had a trial. Have you ever heard of Viadent toothpaste, Viadent mouthwash? This is the company that makes those. They've now been bought and merged, and they're part of Colgate now, I think. But they had a I don't know if you call it a drug, but I guess you would. They inject it down in your gums if you have gum disease, gingivitis, right? And they did a trial where they had a placebo and they had active medication. And they ran the trial, they got all the data cleaned up, got ready to unblind, and the statistician couldn't find the randomization code. So they joked that they had the world's only triple blind study. Even the statistician didn't know. Now, one of the interesting things, here I was a graduate student, an intern, I had no say, no power. The statistician that had lost it was no longer with the company for whatever reason. It's part of why they needed an intern to come in and help, because they didn't have and rehired somebody yet. But I was actually in a meeting with the CEO of that company. They were a small company, so that's not as big a deal as it sounds. But he was always walking around in the hall. And the CEO of the company, who was a PhD chemist, said, well, we'd still be okay, wouldn't we? If we don't have the randomization code, because we can tell who got the drug and who got the placebo just by how they responded. Those that got better must have gotten the active medication. Those that didn't must have gotten the placebo. And I sat there as a student thinking, you got a PhD?
Well, it's in chemistry, so.
Yeah.
That's science.
But the point is, he was a very intelligent, very bright person, and just a lot of people don't think statistically. And I want you to learn to think statistically. Does that bias a study if you just divide your patients by how well they did? Those that did good, you put them in your treated group. Those that did bad, you put them in your placebo group. Are you going to show a difference? Well, of course you're going to show a difference.
So is that what they did?
No.
No.
No. The other people, not even me, the other people are like, no, you can't do that. You can't do that. It wouldn't be kosher. So, and as soon as he thought about it, oh yeah, that was kind of a dumb thing to say. But think about it. If I divide this room in half, and I measure you on some kind of a symptom, and then I divide you based on the value of that symptom, and then do a comparison, of course I'm going to find a difference, right? So, don't ever do a triple blind study.
They never found out, you said.
No, they had to pitch that whole trial and start over. Okay, now I mentioned there several times, and others of you have mentioned the word randomization. Why is randomization important in an experiment?
To prevent bias?
To prevent bias, yeah, okay, exactly. Randomization means you randomly assign people to the different groups, rather than just flip a coin is random. Rather than just saying, I think I'll put you on active and I think I'll put you on placebo. She said to control bias. Now, how does that control bias? think about the biases that could come into play if you let the physician decide who got what treatment. What might a physician do?
Put somebody who needs treatment in the treatment area rather than if they chose them randomly, they would go to the placebo group.
Okay, exactly. That's the main worry, is that the physician will make an assessment of the patient and go, well, this patient's fairly severe. They really need the active treatment. So I'm going to put them in the active treatment group. And this patient's not so bad. They can hold off a while. I'll put them in the placebo group till the study's over, and then I'll get them on active medication. Is that biased the study? Heck yes. You put all the hardest to treat patients in the active treatment. You put all the ones that aren't very severe, don't have bad symptoms in the placebo group. You just bias the study in favor of placebo. So randomization helps control selection bias. Right? That's one thing it does. It also, and just as importantly, balances groups with respect to learning variables. Suppose I do an allergy study, and instead of doing a randomized clinical trial, I just do an observational study where I go down to the local pharmacy and I watch who walks in and buys Zyrtec, and then I ask them if they'll be in a study. And then I watch and see who buys Allegra, and I ask them if they'll be in the study. And then I let them go a week and I measure their symptoms, right? They self-selected into groups that way. Well, what if the direct-to-consumer advertising for Zyrtec is much more effective toward women than it is to men? It makes women want to buy it. It's got like flowers and you know, puppy dogs and stuff in the commercials. I've seen those commercials, right? And then for the males, you know, they're out fishing or whatever, you know, and they're outside, they're out. The Allegra commercials do the fishing and the hunting, and this will make you so you can stand to be outside long enough, you know. You won't be allergic to the thing you just killed, you know, whatever. So the groups end up imbalanced on gender. And a lot of drugs are this way. They work better in males than they do females, or vice versa. So then you've biased your study. Randomization will, by definition, help balance those lurking variables out, even ones that you don't think of. You might not know that smoking affects things, but randomization will tend to put half the smokers in one group and half the smokers in another group. Okay. So randomization is very important to getting a valid result from your study. It's one of the first things I look for when I read an article about a new study. Is I wonder if this was a randomized trial. And if it wasn't, then already I'm thinking, it might give me a hint of something, but it's not going to prove anything. Okay, I'm going to come back to phases in drug development. And this is true in a lot of areas besides drug development, but particularly in drug development, there are 4 distinct phases. The first phase is called non-clinical. And then we have phase 1 clinical, phase 2, and phase 3. Non-clinical means it's not in humans. So it might be in vitro in the lab. You know, does the drug bind to a certain receptor like we think it does, or you know, whatever. Or it's most likely non-clinical phase will contain quite a few animal studies. And a lot of times the animal studies are rats and mice, whatever. The Food and Drug Administration will not allow you to put a new chemical into humans unless you've shown it can go into animals and not kill them, right? Or make them go blind, or you know, whatever. There's no severe side effects when you give it to animals. And they'll take rats and they do dosing by milligrams per kilogram of body weight, right? And that's how they start in man. If they can give a fairly high dose to rats, and nothing bad seems to be happening, then they'll get approval to go into phase 1. And phase 1 are what we call first-in-man studies. This is the first time you've ever given the drug to a human, and you're going to start at really, really low doses. And then if maybe you give a really small dose to 3 people, and if none of those 3 have any side effects, then you'll get a new 3 people and you give them a little bit higher dose. And you'll keep going up till you hit what's called the MTD, the maximum tolerated dose. They start saying things like, well, I'm getting a headache, or I think I'm going to throw up, or whatever. Those are side effects. Okay? Usually you hit some kind of side effect like that before they just drop over dead. So, being a little over the board here, if you had any suspicion that that would happen, you'd never make it this far. Right? You'd never be allowed to do it. But also in phase 1 are studies that are designed, they're called pharmacokinetic. You don't have to write that down if you don't want to, but pharmacokinetic means what does the drug do once it's in the body? Like once you take the capsule, how long is it until the drug reaches a certain concentration in the blood? How long does it stay in the blood? So you do these graphs where you see it going up and then coming down over time, things like that. Those are PK studies. They're done in phase 1. Those are usually done in healthy volunteers. Somebody this morning asked, what are healthy volunteer studies? Because I heard about one on the radio this morning being conducted by Quintiles. Heard of Quintiles? They're over in Lenexa. And what I call healthy volunteers is another way of saying college kids. Right? And because that's where they get a lot of college kids in those studies. And they're people who don't have the disease. They're just testing to see are there bad side effects of this drug. And a lot of times they'll do blood draws on these people and they'll test to see is it decreasing the level of this enzyme or this hormone or something, whatever. If things look good in phase 1, you move into phase 2. These are very small studies. So it's like the one we looked at. It might not have been a perfect design. It was a small number of trials, didn't have a control group. You're looking for any hint of efficacy. Efficacy means the drug works. You're looking for a hint of it. If it looks like, yeah, it seems to be doing what we think it's supposed to do, then you get approval to go into phase 3. And you usually go out to Washington. You have a meeting at the Food and Drug Administration called an end of phase 2 meeting. And you present all your data from your earlier phase studies, and they give you approval. Okay, now you can go to phase 3. Phase 3 are large clinical trials designed to show statistical significance. And that's one of the last terms we've got to get out of this section. Statistical significance. What do you think that means?
So, like causation, I guess, kind of?
Okay, demonstrate causation, yeah. Here's a quick illustration of what statistical significance means. Let's suppose I randomly divide the people in this class into 2 groups. And I'm going to do it by right down the middle, like we talked about earlier. Right, so people in this half are in one group, people in this half are in another group. You don't get to be in a group because you're in the middle. Just kidding. And let's say I took each of your heart rates. Right? Everybody took their resting heart rate, like we did for our survey. And then I compute from those 2 groups the mean heart rate in this group, the mean heart rate in this group. Will they be the same? No, they're not going to be the same. I guarantee they won't be exactly the same. There's no way. But they shouldn't be real far apart. The difference between the groups, what's the only explanation for there being a difference in those 2 groups? Random chance as to what individuals ended up sitting on that half of the room, right? Random chance. So if the difference between groups is small enough that just random fluctuation could explain that much difference, then it's not statistically significant. Now suppose instead of just having each of you sit down, I make this half of the room stand up and do 10 jumping jacks, and then I take their pulse. Now do you think there'll be a difference? The other group stays seated. Yeah, now there'll be a difference, but it'll be a big difference. bigger than the difference when they both did the same thing, which is just random chance. So once you get a difference that's bigger than can be explained by random chance, we say it's statistically significant. Now right now, at this point in your statistical lives, all you have is the eyeball test. Wow, that's a big difference, and the standard deviations are pretty small, so I think that's significant. But later, when we get to chapter 7 and 8, we'll do statistical hypothesis tests called significance tests. And we'll put probabilities on things and say, well, the probability that this is just due to random chance is pretty small. So then we say it's statistically significant. Okay? Everybody know what significance means? All right. You got homework over 4.2 due tonight by midnight. And you will have that margin of error thing as one of those. So this homework we do Friday night. See you then. Turn that on.